Welcome to Part 2 of Lab 5, Protein Synthesis. In Part 1, we discussed the process of transcription, the process whereby the information in DNA is converted to RNA. In Part 2 of the podcast, we'll take a closer look at the RNA produced during transcription, discuss what the information in that transcript means, and describe how ribosomes use that information to make a protein and the process of translation. All three types of RNA are produced by the transcription process we described in Part 1 of the podcast. So messenger RNA, ribosomal RNA, and transfer RNA are all made in the same way. However, only messenger RNA is used as the template to make proteins. So from this point forward, keep in mind that we're referring to messenger RNA. Remember that transcription happens in the nucleus, and afterwards the RNA that's produced exits the nucleus through the pores in the nuclear envelope and enters the cytoplasm. Before it exits the nucleus, though, messenger RNA undergoes a series of modifications. To understand why these modifications are necessary, we have to talk in more detail about genes. Remember that the cells that comprise our bodies are eukaryotic. In other words, they have a nucleus. The genes of eukaryotes have a unique arrangement. Take a look at the figure on the screen. This figure represents a typical eukaryotic gene. At the beginning of the gene, colored in blue, is the promoter, which marks the beginning of the gene. In eukaryotes, each gene contains functional segments, in other words, pieces of DNA that contain the information we need to make a protein, and scattered among those functional pieces are sections of junk DNA, pieces of DNA that we have to remove before we can make a proper protein. Notice that beyond the promoter, some of the gene segments are light green and others are dark green. The light green segments are called exons, and these are good DNA. The dark green segments, called the introns, are the junk DNA. Now, a good way to remember which is which is to remember that introns go in the trash and exons are expressed. Now, when RNA polymerase copies the information in this gene, it copies both the introns and the exons. So before we can use the messenger RNA produced as a result of transcription, those introns have to be cut out and thrown in the trash. There are also two additional modifications that must take place. So let's take a look at how this all happens. During transcription, RNA polymerase makes an exact copy of the DNA. It doesn't know which parts of the DNA template are good DNA and which are the junk, so it just copies everything, leaving us with a non-functional pre-messenger RNA strand. The first thing that happens to that RNA strand is that a protective cap and tail are added. The 5' cap is made of a guanine nucleotide with a couple of additional phosphate groups attached to it, and the tail is made of a string of adenine nucleotides. The cap and tail are important because they protect the ends of the RNA strand from losing important information that's needed to make a protein. After the cap and tail are added, a special set of enzymes called spliceosomes read the pre-RNA strand and cut out the introns and throw them in the trash. The exons are then stitched together, forming a functional RNA molecule that can then leave the nucleus and enter the cytoplasm, where it can be used to make a protein. So at this point, we've talked in detail about the process of transcription. And remember that during transcription, an RNA copy of the DNA is made, and this RNA copy is complementary to the DNA. So on the bottom of the screen is a short DNA sequence. At this point, what I want you to do is pause the podcast and take a few minutes to copy the DNA sequence on the screen. After you've copied it, write the complementary messenger RNA sequence below it. Afterwards, we'll talk about it. Now, I want to urge you not to skip this exercise because this is something that you'll have to do in the future on a test or quiz. So take a few minutes and see if you can figure out what the base sequence would be on a messenger RNA strand produced using this DNA strand as the template. Okay, let's see what the complementary RNA sequence looks like. Remember your base pair rules. C and G are always paired, A and T are always paired, and where you have an adenine in DNA, the RNA contains a complementary uracil. The messenger RNA sequence is essentially one big long genetic sentence, and each of the words that makes up that sentence is 3 letters long, and each 3-letter word is called a codon. Now, just like all of the words in the English language, each of these 3-letter genetic words has a definition, and the definition of each codon is an amino acid. Remember that amino acids are the building blocks of proteins. Also, just like sentences in the English language, the genetic sentence has a beginning and an end. The beginning, or the first word, in all genetic sentences is the same, and that first word is the base sequence AUG, and this sequence is called the start codon. So when looking at the messenger RNA sequence, you first have to figure out where the sentence starts. So you have to scan the sequence from left to right until you find the sequence AUG. So again, here is our start codon. Okay, do not make the mistake of assuming that the sentence begins with the first base. Okay, you may have extra bases at the beginning of the sentence. After you locate the start codon, you should look at the rest of the sequence and mark off each set of 3 bases until you reach the end of the genetic sentence. There are 3 base sequences which serve as the period at the end of the genetic sentence: UAA, UAG, and UGA. So in this particular RNA molecule, we eventually encounter the sequence UAA. This sequence is known as the stop codon. So once you've made some sense of the messenger RNA transcript and you've identified the beginning and end of the genetic sentence and all the genetic words between them, now we have to somehow translate the sentence, and to do that we need a genetic dictionary which looks like this. The genetic dictionary contains the definition for all possible genetic words we might find in an RNA transcript, and it's really easy to use once you get the hang of it. So let's do a quick example. In the messenger RNA transcript we looked at, one of the words in that transcript was UAC. Okay, to find UAC in the dictionary, start at the left of the table and locate the first letter in the sequence, which in this case is U. Then move to the top and locate the second letter, A. Finally, look at the right and find the third letter, C. When you do that, finding the word and its definition is really easy. In this case, notice that the definition of the base UAC is the amino acid tyrosine, abbreviated TYR. Before you move on, take a few minutes and practice using the chart. The figure on the right shows us in a simple outlined way what happens in the process of protein synthesis. Okay, the information in DNA is copied to messenger RNA, and that messenger RNA is translated into a sequence of amino acids that becomes a functional protein. So we've talked about what happens overall in the process of protein synthesis, but there are still several questions we haven't answered. For example, what in the cell is responsible for translating the genetic sentence of RNA? And how does the ribosome actually make a protein? To answer these questions, we have to talk about the details of translation, and we'll begin by looking at the molecules responsible for translating the base sequence of RNA, transfer RNA. Transfer RNA molecules are responsible for translating the genetic code. Okay, the figures on the screen show you two representations of a transfer RNA molecule, but the one on the left is a little too abstract, so we're going to focus on the cartoon figure on the right. The amino acids that are linked together to form a protein are attached to different transfer RNA molecules. So it's the transfer RNA that brings the amino acids to the ribosome so they can be connected to form a protein. But how does the transfer RNA molecule know which amino acid to bring? In other words, how does it decode the message in messenger RNA? Notice at the bottom of the transfer RNA molecule there's a 3-base sequence called the anticodon. The anticodon is complementary to each of the codons that makes up the genetic sentence of RNA. So transfer RNA knows which amino acid to bring to the ribosome by the binding of its specific anticodon to the codons in the messenger RNA sequence. For example, the first codon in all genetic sentences is AUG, okay, the start codon. The complementary tRNA anticodon sequence is therefore UAC. The amino acid attached to a transfer RNA molecule that contains the anticodon UAC is methionine, and methionine is the first amino acid incorporated into every single protein that's made in your body. But remember, we said it's the ribosome that's responsible for actually making the protein. So let's take a closer look at the ribosome. Remember, we talked about ribosomes during Lab 2, and I mentioned that the structure of the ribosome was a little more complicated than how we described it, and in looking at the figures, you can see why. Okay, we're going to focus on the cartoon picture that's in the center. Okay, the ribosome is composed of two separate parts. The larger upper part is called the large subunit, and the smaller lower part is the small subunit. Okay, when ribosomes aren't translating proteins, these two pieces exist separately in the cell. They only come together when they begin to make proteins. Notice in the large subunit that there are two spaces labeled. One is called the A site, and the other is called the P site. Okay, the A and P sites are places where transfer RNA molecules will move into the ribosome, bringing their attached amino acids so they can be incorporated into a protein. So now let's talk about exactly what happens during the process of translation. Just like transcription, the process of translation is divided into 3 parts: initiation, elongation, and termination. During initiation, there are several important things that happen. First, the messenger RNA strand produced during transcription binds to the small ribosomal subunit, and once this happens, the first transfer RNA molecule carrying a methionine amino acid binds to the start codon. At this point, the large ribosomal subunit attaches to the small subunit, sandwiching the messenger RNA between them. Also notice that in part 2 of the figure at the bottom of the screen that the first transfer RNA molecule occupies the P site in the large subunit, leaving the A site free. Now this is important to remember when we begin seeing what happens during elongation. Focus your attention at the top of the figure, and as we continue, follow the direction of the arrows. During elongation, the ribosome moves along the messenger RNA strand like a train on a track, one codon at a time. The free A site allows a second transfer RNA to move into the ribosome, bringing in another amino acid to be incorporated into the growing protein. Okay, if you compare the figures you have in your handouts with what you see on the screen, you'll notice that in the handouts within the ribosome there's a third site labeled, called the catalytic site. The catalytic site is responsible for catalyzing the reaction that links amino acids together. Notice in the figure on the screen that the amino acid chain that's attached to the transfer RNA in the P site is detached from the transfer RNA and then connected to the amino acid on the transfer RNA in the A site. The ribosome then shifts over to the next codon, causing the transfer RNA in the A site to move into the P site and forcing the transfer RNA that was in the P site to exit the ribosome. Okay, this then reopens the A site, allowing for another transfer RNA to move in, thus allowing the cycle to continue. Termination of translation occurs when the ribosome encounters a stop codon. Remember, the three stop codons are UAA, UAG, and UGA. Okay, there are no transfer RNA molecules with anticodon sequences complementary to the three stop codon sequences. So in other words, there's no amino acid that corresponds to the stop codon. So if there's no amino acid to incorporate into the protein, the translation machine comes to a halt and then breaks down. When this happens, the protein attached to the transfer RNA in the P site is released, the small and large ribosome subunits break apart, and the messenger RNA strand is released. Okay, at the end of this process, we're left with a functional protein. The final figure here is a summary of the entire process of protein synthesis. Take a few minutes, look at the figure carefully, and read through the descriptions of this process before you move on. Okay, that concludes this week's podcast on protein synthesis. Now, I know we've covered a lot of material here, and being so abstract, I know that this process is difficult to visualize. So to help you better understand this process, I've provided a link to a really good YouTube video on protein synthesis. Okay, now the video is rather lengthy. It's about 15 minutes long, but make sure you watch all of it because, again, it's really good. If you read through some of the comments people have left, someone described it as a bunch of hippies running around a football field, and that's a pretty good description. Now, it was made back in the 70s, so take from that what you will. But again, I think watching the video will help you to better understand everything we've discussed today.